All right, guys, so this is going to be material for Lab 3. To start off, I wanted to remind you all that you do have a lab practical coming up, so you need to start studying now. The complete list of taxa is on Hayden McNeil, but it's also on Canvas, so you can access it either by going to Hayden McNeil and going to Table of Contents and clicking on the introduction here, click on the introduction of the ebook, and then on the table of contents side, it should say practical exam overview, and it'll have the entire list of taxa there. Primarily, this exam is just going to be images, and we are going to ask you questions about each of the taxa that we talk about. So far, we've covered 4 taxa. We've covered the bacteria. There are the 4 groups that you'll need to know so far. But this week, we're going to also cover some protists. And then next week, we're going to cover protists and fungi this week. And next week, we'll keep going into our mosses and ferns and spore-bearing plants, and then the rest of the plant kingdom. But remember, all the questions are randomized. Not everyone's going to have the same questions. So your practical is going to be during the week of March 8th. I'm going to open the practical at the start of the week, and you'll have the entire week to sit down and complete it in one sitting. You cannot start it and then go back to it later. So it's, a timed exam. I believe you have 130 minutes to complete it, or 120 minutes, but we'll talk about that more when we get to the test. So just start studying now. We also, if you go to Canvas, I have practical exam study material here for you. You click on that. So we're not taking the animal test yet. The first one is going to be the plant practical. So you can also access the list of taxa there. Remember, we've covered these 4 groups so far. This week, we're going to cover all of these groups and then keep going. We'll eventually get to all of these before the first exam. So ways to study for that, I have a really nice characteristic table here. So I've made this, and students really like this. You can put all the characteristics in, anything that we talked about in the lab PowerPoints or the exercises. It's mostly based off of the lab exercises the exam is. You can also play these Kahoots. We're going to do a exam review, and this is my review PowerPoint if you want to study that. A student created an Adobe Spark page, so you can click on that too, but lots of great resources here. Okay, so that's the practical. Now, for Species Spotlight, you need to submit your group's article. So everyone in your group is going to have the same article, and you all will need to submit that by February 28th. I decided to extend it because you all have a lot of work that's due this week, and we'll talk about that more later. But for right now, make sure your group is talking actively, you're having meetings, and you choose an article before February 28th. I do need to approve it before you can move forward with the project. So make sure to get that in on February 28th or before. That way your group can move forward with their project. Okay, so make sure you also have signed up for a group. If you haven't done this by Tuesday, February 16th, by at least 5 p.m., I would have probably already done it for you, but you'll just go here, Species Spotlight topic sign up, and click the edit button at the top and just type your name in to whichever group you'd like to work with. If your group, everyone should have enough people, at least 3 to 4 people to a group. If not, I will contact you and we'll work it out. So after your group finds an article, individually, you're going to be writing a paper. You're going to need to submit a summary of that paper to the Writing Center by March 7th. You're also going to have to upload evidence that you attended the Writing Center or an online appointment by March 7th. You're going to have to upload that to Canvas. So you're going to have to send me a draft of your paper, and you're also going to have to send the Writing Center a draft of your 1-page paper, single spaced. Okay, and all the information is on Canvas as well for this. As a group, you're going to be creating an Adobe Spark web page. If you need help like signing in or anything, you're going to sign in to Adobe Spark using your UNCW information. You'll have to sign in as Enterprise on there. If you have trouble creating an account or anything, just contact me. And that is due April 4th. So that's the very last thing you're going to do. Your paper is also due on April 4th. So this needs to be done early on. This is just by the end of the semester, pretty much. And don't forget that everything for this project should be cited in APA. So this is the use of an in-text citation. This is the full citation that you're going to need in your bibliography. So the answers to Lab 2 exercises are here. Go through these and make sure you update your last week's lab. Again, when I grade labs, I'm primarily doing it based off of completion. The information, if it's absolutely incorrect, that's when you start getting points off. But I can definitely tell after teaching this for several years if you tried or not, or if you cheated or not. So please make sure to update the answers. That way you can study for the lab practical in the next couple weeks. So here are the answers to the second page. And difference between selective and non-selective media, definitely talked about that, so don't get this wrong. Differential, remember it does allow you to differentiate between species, but you also get this color indicator. So change in color can also help you differentiate species on a plate. So now we're going to talk about protists. So what is a protist? Well, this is a paraphyletic group. So it has multiple ancestors here. We primarily have unicellular organisms, some multicellular organisms. For example, algae are multicellular. Protists can be fungus-like, animal-like, or plant-like. So there are 7 supergroups, okay, that you're going to need to know. Remember, all of these belong to domain Eukarya. So they have those membrane-bound organelles, they have a nucleus, things like that. So we're going to start by going through each group individually. So excavata, this group has multiple different kinds of protists here. Primarily their synapomorphy, or the trait that unites all of them together, is that they have a feeding groove. These organisms can be autotrophic, heterotrophic, or parasitic. So in the case of Euglena, these are autotrophs, so they do create their own food, but they also are heterotrophic, so they can eat nutrients in order to gain energy. We also have this organism Trypanosoma brucei. It's a parasite, and it causes African sleeping sickness. Giardia, which is a horrible parasite that you can get from drinking contaminated water, like in a creek or a river, causes horrible intestinal problems, vomiting, diarrhea, things like that. So those 3 organisms are members of this group. So you should know, the synapomorphy for the group and a little bit about the group. So these are the kind of traits that you could write into that characteristic table. So these are the species that you're going to need to draw. Remember, on this slide too here, the Trypanosoma, these are red blood cells in the back. These are the Trypanosoma. So that's the parasite here. These are just red blood cells. So again, here, these globs are the red blood cells, and then the purple is the actual parasite. Here's Euglena, 2 images of Euglena. You can draw either one. You could draw this one here and label it if you'd like. But make sure you do that. It's part of your lab exercises. So the next supergroup is Archaeplastida. These guys have a shared ancestor with plants. They're primarily multicellular. So almost everything in this group is algae. And the trait that unites them is chlorophyll A. So the ability to photosynthesize using that pigment is really important to this group. So we have organisms like Volvox and algae in this group. If you're looking at a Volvox, this is the entire cell, but inside you have what are called daughter colonies. So these circles, your lab manual is going to ask you to label them. So make sure you label the inner circles as the daughter colonies. Again, red algae is where we get our sushi from, like nori. So these are also in this group. And then there are green algae and seaweeds are in this group as well. So the ones that you need to draw here, the daughter colonies are on the inside, so label those. And then the outside, okay, is the whole entire organism. Here you have Polysiphonia, and you'll need to label the tetraspores, which are on the inside here. They're just spores in groups of 4, right? So then we have what's called the SAR clade. So this clade or group consists of 3 supergroups. So you'll need to know a little bit about each of these 3 supergroups. So the first group, Alveolata, has a couple different types of organisms in it, but the trait that unites them is called alveoli, and these are little air sacs for locomotion and movement, feeding. So we have ciliophorans like paramecium. They're going to be covered in cilia, which helps them eat and move around. Then we have apicomplexans that are primarily parasitic. So we have this organism called Plasmodium falciparum, and that is what causes malaria. So again, when we're looking at a blood smear like this one, these purple circles in the background, those are our red blood cells. But the plasmodium is this little kidney bean organism here. So make sure you can label that properly. But plasmodium is transported by mosquitoes, which we know. So that is how people are getting malaria. This little parasite is inside of that mosquito's blood and then is being transferred to another host, for example, a human, right? So then we have dinozoa. So these are our dinoflagellates here. They have this really strange structure. They also have these things called theca plates. And so if the organism is thecate, it has the plates. So dinoflagellates are thecate typically. And the plates are just like a little armor, like little shield that they have. Okay, so these are the species that you'll need to draw. The serratium and then the paramecium. And I don't think you'll need to label them. Just make sure the dinoflagellates are thecate. Okay, so if it was athecate, does not have the plates. If it's thecate, it has the plates. All right, you'll also need to draw plasmodium. Make sure you label the red blood cell versus the little plasmodium. Our next group, arguably my favorite one, is Straminophylla. And you have a couple different types of organisms, different protists in this group. The first one we're going to talk about is diatoms. So they come in all shapes and sizes. You'll need to draw some mixed diatoms. And their cell walls have silicon dioxide, which again is like glass. It's very abrasive. So abrasive that we put it in our toothpaste and other household products. Some people actually eat this stuff. It's actually great for getting rid of household pests as well. But you'll need to know that they're photosynthetic, okay? And they have little hair-like projections on the surface of their flagella. And primarily the biggest thing, they're photosynthetic and they have silicon dioxide. They're abrasive. So here are some mixed diatoms that you can draw. Again, they're different shapes, so maybe just draw a couple different shapes. You can take some from here or either of these micro slides. The next group of organisms still within Straminophylla here are our kelp. So think, Harry Potter, our kelp forest. These organisms are multicellular, photosynthetic, and they have a pigment that gives them the brown olive color. It's called fucosanthin. So that is the word you're going to write in your lab exercises. But it asks you what kind of pigment gives it the brown olive color. You're going to need to draw laminaria. This is the species here. You can draw it on its side if it fits better into your manual. And then the last group within Straminophylla are our water molds. These are heterotrophic organisms. They're actually responsible for the Great Potato Famine in Ireland, specifically P. infestans. That was the whole problem there. You also get some issues in aquarium tanks. So yeah, these organisms are super important as well. Our next supergroup we're going to talk about is Rhizaria. So there are 2 main organisms in this group. You will be responsible for knowing the names of these. So within Rhizaria, you do need to know these 2 names. So we have silicon dioxide based organisms. These are our radiolarians right here. So remember, silicon dioxide is pretty much like glass, right? If you see this word test, it just means mineral skeleton. So its skeleton's made up out of silicon dioxide. But notice, remember that these are like glass, right? And they kind of look like glass. They are see-through and they don't have much coloration to them. Our foraminifera, these guys are made out of calcium carbonate. But what else is made out of calcium carbonate are seashells. So these guys actually kind of look like seashells. So on this, they're also, responsible for the White Cliffs of Dover, making them so white, because calcium is very white. But notice here, this is a better picture. This is one I was kind of referencing to. You can see that they look like seashells, and they are also made out of calcium carbonate. These guys, silicon dioxide. All right. Our next to last group here, amoebozoa. These guys are heterotrophic. We have some interesting organisms in this group. We have slime molds, which are like nature's natural little GPSs. And then we have amoebas. So these organisms eat by phagocytosis. Remember, everything in this group is heterotrophic, so it needs to eat something else to gain energy. But with phagocytosis, they have these little pseudopods, these false feet, right? And they're going to wrap those feet around their food and then slowly digest it over time. So you'll need to draw an amoeba. You can, I don't think it requires you to label anything. Just make sure you draw it. And then our last group that we're going to talk about is Apistoconta. So this is a very weird supergroup because it actually contains kingdom fungi and kingdom animalia. So as far as taxonomic ranking, supergroup is kind of a weird word. It's above kingdom. It's kind of in the middle of domain and kingdom. But it's really just a little addition here to kind of try to help us classify things. But in this group, we do have fungi, which we're going to talk about next. But we also have these little guys that we'll see when we talk about animals again. They're called choanoflagellates or collar cells, right? Because they have these little collar cells. But they live inside of sponges primarily. But we'll talk about them more later. So right now, we'll stop here and you'll watch another video on fungi. So you'll have to do the lab exercises for Kingdom Protista. There's also a pre-lab homework assignment that you'll need to watch this video and make sure to submit this with your lab 3 information, okay? So really at the end of all this, we have a lot going on this week. So you'll need to sign up for a group. Your group needs to meet and figure out an article. That's due on Canvas by the 28th. I won't open that until tomorrow when all of the groups are finalized. You have 2 quizzes to do. So you'll have one quiz on protists, and you'll have another quiz on fungi. Then you have to submit these lab exercises. You'll always submit lab material on Canvas. You will never submit lab exercises on Hayden McNeil. The only thing you're doing on Hayden McNeil are quizzes and accessing the ebook. So you'll submit the exercises for the protists, which are pages 15 to 19 in your lab manual. And then after you watch the fungi video, you'll need to submit information for that's located on page 37 in your lab manual. So we're kind of going ahead because it's a little out of order here according to the way we're doing it this semester. And then you have that pre-lab assignment. So I need you to submit all of this together, okay? So this page, all the protist pages, and then page 37, which is the fungi, okay? And that's all going to be under lab 3 submission on Canvas. So when you're on there, make sure, again, this should be your view of Hayden McNeil. You have 2 quizzes, right? You don't need this because you should have the lab manual already. If you do, I have blank pages as well on Canvas, but I haven't opened the assignment yet, but I'll make sure to put detailed instructions for lab 3. Okay, so you'll submit all of that on Canvas. All right.